Kia ora, everyone. My name is Xavier Pochon. I work at the Courtron Institute and University of Auckland. I am honored to be able to present this decade-long project, which would have not been possible without the very precious help of all of these wonderful people listed here. I would also like to thank Jan Pawlowski, Thorsten Stock, and their research groups for valuable input throughout this project. Salmon aquaculture is the fastest growing food production system in the world, now accounting for about 70% of the market. In New Zealand, the salmon farming industry is a lot smaller than in other countries, for example, Norway or Chile, but it is a growing sector and its production does impact the surrounding environments and, in particular, the benthic ecosystems just below the farms due to high organic loading from fish feces and excess feed. And this impact has to be monitored by law under the Resource Management Act. Many indices have been used internationally for measuring the degree of benthic impacts around salmon farms, but until recently, it was not clear which one should be used in New Zealand. As you can see, salmon farming is a relatively recent business in New Zealand, and it was only in the first decade of the 2000s that the increase in aquaculture space fueled intense public and governmental scrutiny, which led to the formulation of the Enrichment Stage Index, and I will describe this index in the next slide. This also led to an important legal trial through the Environmental Protection Authority that justified the need for the implementation of the benthic standards working group. This is a relatively small group that includes representatives from key government agencies, science providers, industry, and community stakeholders. It is this group that created, maintained, and update the best management practice guidelines that guide monitoring efforts under the resource consent conditions set for high and low flow salmon farms in the Marlborough Sounds. And this is the region in the top of the South Island where most of the salmon farming activities are taking place. The Enrichment Stage Index is quite unique as it incorporates both biological and physicochemical metrics in one index. This method involves the collection of large amount of sediments along the enrichment gradients from controlled sites and at regular distance intervals all the way to the fish pens. And microscopy is typically used to measure changes in macrofaunal distribution along that gradient. The index is a quantifiable bounded continuum or scale of impact that ranges between 1 or natural state to 7 or highly organically enriched, and along these gradients exists a range of compliance thresholds that are detailed in the management guidelines and which may vary depending on the farm resource consent conditions or on the water flow environments where the farm is situated. This method works really well and it is the gold current standard, but the fundamental problem is that it is costly and time consuming. It typically take about 4 to 5 months from collection to reporting until any action can be taken, and this, of course, is problematic for everyone. So about 10 years ago, we identified eDNA metabarcoding as a very promising alternative to microscopy for some of the obvious advantages that are listed here. But in particular, the time and cost reduction was extremely appealing, but also the potential for standardized processing workflows between distinct laboratories. And so to do so, we set out 3 aims. First was to develop a metabarcoding-based biotic index that could eventually replace the microscopic-based macrofaunal components of the enrichment stage index. Second was to improve protocols and automatic data processing. And third was to refine and validate the index so that it could be incorporated into the best management practice guidelines for routine compliance monitoring. And just to give you a little bit of timeline to that, project started in 2012 thanks to a Ministry Business, Innovation and Employment funding, which allowed us to explore benthic foraminifera over a couple of years. We also looked at spatial temporal patterns and various other aspects. This led to an additional funding, Seafood Innovation Limited funding, which is a conglomerate of government, private, and industry funding. And that led us to look at other indicators, and we looked at total bacteria and also at eukaryotes as other indicator and combined. But really, this exercise showed that bacteria are really amazing and outperformed any other indicator. So we presented in 2017 in front of the Benthic Standard Working Group a preliminary bacterial-based metabarcoding biotic index. This shows the sampling locations on the South Island of New Zealand and the sampling effort conducted over this 9 years project. 12 fish farms, 60 stations, a number of regions and subregions, 1,000 sediment sample collected, and 400 that were absolutely fully analyzed and compared between both the traditional microscopy-based methods and the novel method. And of course, we made sure that the metabarcoding method would be systematically confronted and compared to the traditional method for consistency. And as mentioned earlier, bacterial communities really outperformed any of the other bioindicators that we looked at, and therefore the rest of the talk will focus on bacteria only. So how did we calculate this metabarcoding index? It first started by using regression spline analysis, which identifies the key bacterial ecogroups or taxa or species that consistently peak over a given enrichment stage. And these ecogroups are only retained if they show consistent ecological preferences for these categories over multiple farms, multiple years, and flow environments. The ecogroups are then stored into this precious gene code ID database, which is used as a reference to interrogate new data. Then, the proportion of these different ecogroups are used to calculate a presence-absence-based bacterial metabarcoding biotic index, which ranges on the same scale as the traditional ES. And really what we want to see here is a perfect relationship, a one-to-one relationship that enables us to get the same scoring for compliance. And again, bacterial indicators were amazing. They showed really strong bacterial shifts in their community composition from anoxic conditions near the cages and across into more natural seabed conditions. And this was really consistent across years, farms, and flow environments. Through this exercise, we were able to isolate approximately 400 key bacterial ecogroups into the gene code ID database, and the preliminary index was really promising with very strong regression values, regardless of flow, as you can see here. But you can also see that it was not a perfect relationship, and we were somehow lacking some of the bacterial indicators at the high end of the distribution, something that will be addressed later in this talk. Those preliminary results were really well received by the Benthic Standards Working Group, but the general request at that time was that more data should be gathered, protocols should be improved, and automatic pipelines should be investigated to take the tool one step closer to implementation. And that's what we've done over the next 2 years. So the next couple of slides will show a bit of a laundry list of some of the key parameters that we used in our protocol. The first point I wanted to make is that we used a Van Veen grab sampler throughout this project, and it is well acknowledged that this is not an ideal method for eDNA, but we had to do so because we were comparing with the traditional method, and we needed to also collect the macrofauna. So if you can, it's better to use other type of cores, box or cylinder cores. The Van Veen grab were brought to the surface, and about 5 grams of the top 1 to 2 centimeter layer of the sediment were isolated throughout the grab, and then isolated on ice and in the dark for a few hours until we got back to the laboratory where they were stored at -80 C. So other grabs are better, I just said that, but there are a number of interesting developments with new sampling devices for eDNA, and I'd like to just highlight 2 of those. The first one is the substrate-independent Benthic sampler developed in Tromsø, Norway by Dr. Nigel, Keeley, and colleagues, which is a device that enables the collection of bacterial communities from organic flocks deposited on very deep reefs and that are essentially rocky reefs. Works really well. The other one exciting device is the syringed ROV developed by Sean Robinson and his colleagues at DFO Canada, which is a little ROV that you can manipulate and even bring down under the fish farms, and it can very effectively collect eDNA from mats, from soft sediments and encrusting surfaces. Second point I wanted to make is that if you are intending to work with DNA and RNA, it is much better to snap freeze your samples or use isolation buffer instead. For extraction purposes and anything in the lab, first thing you need to make sure is that your lab is well equipped, is a dedicated molecular laboratory with appropriate QA/QC protocols in place. It is recommended to use commercial DNA extraction kits, which allow for some sort of standardization between analyses. And in this recent paper by Pearman and al., we looked at a range of commercial kits and amount of starting material from 0.25 grams, 2 grams, and 5 grams, and could show and demonstrate that the QIAGEN PowerSol kit is the most effective, provides the best quality and quantity of DNA, as well as highest bacterial richness compared to other kits. And the beauty of this kit is that it can be automated using robots, liquid robots in the lab. Also, very importantly, it is extremely important to add extraction blanks throughout the process, because there will always be some background contamination of bacteria within the kits. Over the years, we consistently targeted the exact same V3, V4 region of the bacterial 16S. Of course, again, it is very important to include the appropriate extraction blanks, PCR, and sequencing controls directly into the library that's been sequenced. We looked at a range of library preparation techniques, but we now are very happy with dual index sequencing and the use of 500 cycle chemistry kits of Illumina. A few years back, we moved into de novo processing using Data2, which generates ASVs or amplicon sequence variants, which are infinitely better than OTU clustering. We typically remove samples that have less than 10 to 20,000 high quality reads per sample, and we also remove ASVs with less than 10 reads per data set. Regarding automation, we developed a automatic R-script pipeline that now enables automatic regression spline analysis, automatic eco-group selections, and automatic calculation of the metabarcoding biotic index. And this is really a game changer for us. It really enables the generation of the index in a click of a button, which of course allows a great time and cost savings in the computer pipeline. Following these improvements, we met again with the Bentic Standards Working Group in 2020. The group was really pleased with the developments and was inclined to incorporate the new tool into the best management practice guidelines, providing final index refinement and validation against compliance thresholds analysis. And that's what we've done. For the index refinement, we exclusively focused on the Marlborough Sounds region, where 9 salmon farms are currently in activity. And you will remember this regression analysis that I showed you earlier, not a perfect match, but still very good, in particular when considering that this incorporates all 9 farms, yet it was calculated using the original gene code ID database, which was represented by only 3 of those 9 farms. So we built onto this original data, we updated it a bit, and looked for additional key bacterial eco-groups, particularly those representative of the high end of the spectrum here. And we also improved the BMBI index by adding an additional eco-group class 6. And finally, we calculated an overall molecular ES, which was based on the same weighting as the traditional index in terms of physicochemical and biological parameters. And that is 10% of organic loading, 20% of sediment chemistry, and 70% based on the BMBI. And the BMBI here essentially replaces the macrofaunal components of the original equation. And as you can see, this really improved the results, here shown at the station averaged level. So each dot here represents an average of the triplicate samples collected at each station. We now have a near perfect relationship between the two methods, regardless of water flow environments. And the ES scores really mirror each other and across the enrichment gradient, which is exactly what we want for this compliance threshold analysis. For this threshold analysis, we compared the mean ES scores obtained with both methods across the 9 years periods. And this is just a subset example, only showing 5 farms that are located in a high water flow environment. You see the years here, and these numbers represent the distance in meters to the fish pens. And those dotted lines here represent some of the proposed thresholds for compliance in high flow environments as described in the new management practice guideline document, with the mean ES scores expected to fall below or within these thresholds. As you can see, the mean ES scores obtained with both methods were very similar to each other, and in fact the molecular index correctly classified station compliance in 62 out of the 67 stations surveyed through these time periods. And for the 5 that did not, for example, these 2 here, they were very close to each other and fell on either side of the line. The take-home message here is that the analysis of station average ES score showed a very high level of agreement between the molecular and macrofauna-based indices, and that the mean ES scores differences between the methods throughout those 9 years and 9 farms were very minimal, with only 5 stations showing differences of more than 0.5. The other take-home message is that the confidence intervals values around the mean ES scores are approximately half those typically obtained with the traditional macrofaunal based index. And you can see here an example showing obvious differences in confidence interval values for the traditional index. And this indicates the new index is less variable when measuring enrichment levels within a given sampling station. And it also suggests it is less influenced by the spatial patchiness that can be evident in macrofaunal assemblages. And therefore, this is really a key advantage of the molecular-based index because it can provide more certainty around the results when there are instances of non-compliance. To summarize, this project was able to successfully develop, test, validate, and refine a DNA-based index for benthic monitoring of salmon farming activities here in New Zealand. This has been a relatively long yet absolutely fascinating journey, which was really only made possible through systematic multi-year comparison of both the new index with the traditional index. And so having access to these farms and all of the precious samples and associated data has been really invaluable. And we learned heaps. For example, we gained a much deeper understanding of the diversity, ecology, spatio-temporal distribution as well of a range of taxonomic assemblages that are really understudied here in the New Zealand marine ecosystem. This is true for benthic foraminifera, bacteria, and general eukaryotes. This project also revealed the incredible value of bacterial assemblages as powerful indicators of environmental stress. So going through the research and development process, we were able to improve the sampling methods, to optimize the DNA extractions, and to standardize our protocols. We were also able to generate tailored bioinformatics and automatic index calculation pipelines. And really, the result is a monitoring tool that is significantly faster and cheaper. Now, it's one thing to develop a molecular diagnostic tools. It's an entirely different beast to actually bring it to implementation into the legal framework. And the way we operated was really to work very closely with a range of the key end users and stakeholders from the very early onset of this project and all throughout, which helped us, fine tune and get where we are now. We have now just submitted our final report to the Bentic Standards Working Group for assessment and are awaiting further discussions with them, which will dictate their decision on whether they deem the new tool now ready for incorporation into the best management practice guideline for routine monitoring. And finally, I'd like to acknowledge and thank some of the key people from private, government, and industry sectors that have supported this project through funding, through valuable advice, or international experts' opinions and reviews. And of course, the funding agencies that helped support this project altogether. And thank you for your attention.